we're going to be looking at the ultracellular structure and function. Okay, so we're looking at ultracellular structure and function. In the previous class, we also talked about the ultracellular structure and function, but in this case, we relate it with the usage of the light microscope, how we can get the cell material and then make it into a thin section and then stain it with a dye to be able to study the structure. Scientists were able to use that method to study the structure. Watch the part, I think that should be part 5 of biology. You'll see that. Now, in this section, we're going to be looking at the ultracellular structure and function of the cell using the electron microscope. In this case, what is done is that the cell in question, number one, the cell in question is going to be ruptured. Okay, it will be ruptured. Now, when it's ruptured, it will be able to show the ultracellular structure, meaning the detailed form of that cell. And in doing that, two methods will be carried out. Those two methods, one, what they will do is that they will carry out what we call centrifugation. Centrifugation. What's the essence of centrifugation? The essence of centrifugation is to separate the organelles in the cell into cell organelle. So centrifugation will help separate the cell organelles, so they will be singly. Okay, so the biologists or the scientists will now be able to begin to carry out some biochemical activity. Number 3, biochemical experiments. Okay, biochemical experiments will now be carried out on each of these cell organelles. Now you might be asking, you hear me say cell organelles. What are cell organelles? Cell organelles are special structure that performs a specific function in the cell. That's what we mean by cell organelles. So a biochemical experiment will be carried out on each of these organelles. And then this will now help scientists, number 4, to determine the function of each of these organelles. Determine the function of each of these organelles. So that is the essence of doing of carrying out centrifugation, so that the organelles can be separated, and then they can now be studied individually using a biochemical experiment to determine the function of each of these organelles. Meanwhile, chromatography would then be used to identify the chemical substance in the reaction system. So number 5, chromatography will be used to identify the chemical substances in the reaction system. To identify the chemical substances in the reaction system. Meaning what is taking place inside of the cell. For you to know what kind of chemical is released and the reaction that is taking place, chromatography will be used. You know, chromatography is one of the separation techniques in chemistry used in the separation of colors. So take note, go watch my chemistry part 1, part 2, part 3, part 4, part 5, part 6. It's still talking about separation technique as the case may be. Okay, so with this, we've been able to have an idea what the electron microscope will be able to do, so that we can be able to use that to study the function of the cell. So let's start now by describing the functions of the cell. The first we want to look at is plasma membrane or cell membrane. Number 1, we have cell membrane, or we call it plasma membrane. Okay, take notes. That's another name for cell membrane. Now, the plasma membrane, this is the outer covering, of a cell, especially in animal. But in plant, the plasma membrane is covered by the cell wall. Now, number 1, take note, the cell membrane or plasma membrane is an outer covering. Put that in your mind. Number 2, about this cell membrane, it is predominantly made up of protein and lipids. It is made up of protein and lipids. Okay? Protein, fat and oil, okay? And also, this cell membrane, I also want you to know something, that it is also thin. This cell membrane is thin and selectively what? Permeable. I'm going to explain. What does it mean? It means that it can allow substances or material to move in and out. So the cell membrane, another thing again it does that, it regulates or prevents materials from moving out of the cell. So it prevents materials or substances, okay, from moving out of the what? The cell. Can you see that now? So it is thin and selectively permeable. Permeable to what? To ions. Permeable to what? Water. Permeable to what? Ions dissolved in water. Okay? Permeable to different substances that are going to be useful to the cell. Okay. And I also want to let you know that, okay, I've told you earlier on that this cell is found in plant, it is found covered by the what? The cell wall. So it is called the cell membrane or the plasma membrane. So I've been able to look at outer covering. It contains protein and lipid, and it is thin and selectively permeable. I think some textbooks, some materials I read, they say it is a bilaminar layer, okay, that has phospholipids. Okay, I've read in other material, but just take this. These are important. Outer covering, protein, it contains protein and lipids. It is thin and selectively permeable to ions, water, and dissolved substances. It prevents materials from moving out of the cell. That is the cell membrane. It was through the electron microscope they could be able to study and know this cell function. So let's look at the next one. I want to look at number 2, the cell wall. Just follow me now. Follow me, the cell wall. The cell wall. Now, the cell wall is also, number 1, an outer covering. Take note, outer covering. Okay? The cell wall is also an outer covering. Number 2, it contains cellulose. Cellulose. Okay? It contains cellulose. Number 3, it is freely permeable. It's freely what? Permeable. Meaning that it can allow for substances to go in and out. Okay? It is freely what? Permeable. But unlike cell membrane that is selectively permeable, it only allows what is permitted into it or what should go out. It is freely what? Permeable. And I said it contains what? Cellulose. Number 4, it gives protection. Gives protection to the cell. Can you see it now? Number 5, it helps for mechanical support. It helps the plant it gives the plant mechanical support. Okay? It gives the plant mechanical what? Support. Okay? You can see that is what the cell wall does. So it's also an outer covering. It contains cellulose. It's also freely permeable. Okay? Not selectively, freely permeable.